Chapter 11, we're going to just kind of scratch the surface a bit of how, you know. Now, cancer cells are cells that were once normal and now they've been transformed where they grow out of control. What they found with these kinds of cells is that they have found genes that code normally for growth factors being transcribed and translated too often, so that too many growth factors are being produced and the cell, that's encouraging this prolific cell growth. It can also have to do with receptors in the membranes of these cells being produced at too great of a rate, or even some of the intracellular molecules that are involved in some of the signal transduction pathways that overstimulates cell division. Well, a little bit of history of this. They've also found, though, that viruses can actually cause cancer. In 1911, they discovered that there was a particular virus that carries a gene and can actually cause cancer in chickens. And of course, more recently in the news, we've heard about HPV being associated with cervical and other types of cancer and can be transmitted through sexual contact. Well, there was also the discovery, the understanding of something called proto-oncogenes. In 1976, Michael Bishop and some of his colleagues discovered that in this 1911 cancer-causing chicken virus, that this virus actually contains an oncogene. And what an oncogene is, it's just an altered version of a normal chicken gene. And the normal gene does have the potential to become a cancer-causing gene, and because of that, it's referred to as a proto-oncogene. Okay? So, a proto-oncogene is just that. It's a gene that it's a normal gene, but it can become a cancer gene in time. Alright, so once again, a proto-oncogene is a normal gene. But what can happen to a proto-oncogene is that it can experience a change, mutation, and it can become a cancer gene. And at that point, it's referred to as being an oncogene. Now, what can create the change can be just a spontaneous mutation. We looked at mutations earlier. But there can also be interaction with the environment that can encourage these changes. You think about being exposed to, say, chemical carcinogens or X-rays, or in some cases even viruses. What researchers have found is that proto-oncogenes oftentimes are coding for proteins that are involved in influencing the cell cycle. Now, when these proteins, these proto-oncogene proteins, are not misbehaving, then they are producing these genes are producing the right proteins at the right time in the right amounts, and they're regulating cell division just like they should. But let's take a look at our figure we have at the bottom of the slide. Here we have a theoretical proto-oncogene, and it codes for a protein that stimulates cell division. So that's the job of the protein coded for by this gene. What can happen is a mutation within the gene, a spontaneous mutation, can lead it to become an oncogene. And now that change in the nucleotide sequence codes for a protein that's hyperactive. And so that hyperactive growth stimulating protein, it's being produced in the normal amount, but it's hyperactive, and so it overstimulates the cells to divide. Another scenario with this theoretical proto-oncogene is that it can get copied multiple times and inserted within the chromosome. So now there are multiple copies of the gene, and when one gene is copied or transcribed, they all will be transcribed. And so there's a normal level, activity level for each of the proteins, but they're still being produced in excess. So it's a normal protein, but there are too many of them being produced because of, the excess copies of the gene that have been put into the chromosome. Another thing that can happen has to really do with, say, a translocation event, where a gene is moved to a new location within the DNA, and it's next to maybe perhaps a more active promoter. And now what happens is that there will be normal growth proteins being produced, but Now, there are three kinds of DNA changes that can convert a proto-oncogene into a cancer-causing oncogene. And I know we've already talked about mutations a bit, but there can be a point mutation within the proto-oncogene. And like I said, that can lead to a hyperactive growth factor, like we saw in the previous slide. There can also be amplification of the proto-oncogene to where there are just too many copies of the gene within the chromosome, and so normal growth factors will be produced, but there will be too many of them being produced. Or, as I mentioned thirdly on the previous slide, there can be translocation of a gene next to a more active promoter, and that will lead to excessive transcription translation, so there will be too many normal growth factors being produced. So what has happened, of course, is the normal gene expression has been changed, and the cell is stimulated excessively to go ahead and divide. Now, there are also tumor suppressor genes that can be affected and lead to cells becoming cancer cells. Cells do contain tumor suppressor genes, and their proteins that they end up producing are normally involved in inhibiting cell division, and that way, this helps prevent uncontrolled cell growth. But a mutation in these tumor suppressor genes can actually decrease the activity level of these tumor suppressor proteins, and so that may contribute to the development or the onset of cancer in some individuals. So they've also found, though, that some of these tumor suppressor genes are actually functioning in the repair of damaged DNA, and so a mutation in that, of course, could lead to an accumulation of cancer-causing mutations. So that's what's being shown here. Here's a tumor suppressor gene producing a protein that behaves in a normal way to help control or inhibit cell division, to help it stay under control. But if a mutation occurs in that, it can lead to a protein that cannot inhibit. Now, it's not like oftentimes what happens is there will be multiple changes in the genes in the development of cancer. The American Cancer Society estimates that nearly 100,000 new colon cancer cases will occur in the year 2014. So that's why, and they look at the data, and of course that's why, at the age of 50 anyway, it's recommended that you go and you have the colonoscopy done. And of course, in the colonoscopy, they're going to be looking at the large intestine. Now, the development of malignancy is a gradual process, and that makes sense because you think about an oncogene arising. It's going to arise after an accumulation of mutations, and of course, when you have these cells these tissues that do have kind of high rates of cell division. The control mechanisms in those kinds of cells are certainly critical that they continue working exactly as they should. But as there's this accumulation maybe of some or the beginning of say an oncogene, a proto-oncogene becomes an oncogene because of a mutation, then the cells begin dividing at a greater rate. And then what can happen with even more DNA mutations is that there can even be the inactivation of tumor suppression genes, or once again, there is the tumor suppressor protein is not functioning like it should. And so in time, that can lead to the formation of a malignant tumor. And of course, at that point in time, some of the cells can break loose and metastasize and move to different parts of the body. And so oftentimes what's going on is that several mutations are actually having to accumulate within cells, and so this is why oftentimes cancer develops later on in life, is because it takes a while for enough mutations to occur. Now, faulty proteins, we mentioned this a little bit earlier in the chapter, but there can be faulty proteins within transduction pathways, and that's what we're going to look at here. what we have in this case, we have a normal growth factor binding to a receptor tyrosine kinase. We call it an RTK. They're just calling it a receptor here, but it's an RTK. And what this RTK does in this particular case is it activates a G protein, and in this case it's called a RAS, a RAS protein. Okay. Now, where that RAS, the RAS actually comes from, it really stands for rat sarcoma, which is a type of cancer. Now, this activated RAS protein, this G protein, it has a GDP displaced from it, which is replaced by a GTP. That GTP activates the RAS protein, which then activates what's called a phosphorylation cascade, and that's where you have these relay proteins being phosphorylated. So this is normally how this particular system works. So you have this whole line where it reads down here relay proteins. You'll have this whole line of protein kinases, and they're phosphorylating one another essentially. Now, the last kinase here in the signal transduction pathway, or the line of protein kinases, activates this transcription factor, this little purple guy here just outside the nuclear envelope. And when it does that, that transcription factor can then go in and bind to the promoter of a gene, and that's going to basically activate the gene, turn on the gene. Now, what that means when we say turn on or activate a gene, that means the gene will be transcribed. And we've talked about transcription. You know what that is. That's the building of messenger RNA off the DNA template. So the gene gets transcribed, the messenger RNA is produced, and then the messenger RNA makes its way outside of the nucleus into the cytoplasm for translation. And then once translation occurs, that produces a protein, and that protein's involved in stimulating, getting the cell to go ahead and divide. Now, what can happen is that there can be a mutation to the RAS gene so that it produces a hyperactive RAS protein. So that guy here is no longer like this, but the mutation creates a hyperactive RAS protein like we see here. And what that means is this hyperactive RAS protein will keep stimulating this signal transduction pathway, which stimulates or activates this transcription factor, which turns on the gene and tells the cell to divide. And so that can lead to, increase. So what we're looking at are faulty proteins. And so we just looked at, a faulty protein in terms of a RAS protein. Now we're going to look at another faulty protein that happens to be a transcription factor. So what we have going on in this diagram is we have a normal growth inhibiting factor represented by kind of this yellow triangle binding to a receptor, to a plasma membrane receptor. Now that in turn leads to activation of a transduction pathway represented by these teal colored drawings. These are proteins making up a signal transduction pathway. Now, what the last member of the signal transduction pathway does is it activates a transcription factor. That's called a p53 protein. Now, that p53 protein is basically a tumor suppressing protein, or it's a protein product. Now, if you notice what the activated p53 protein does, it's a transcription factor. It's going to bind to a promoter, and that enables the gene to be copied. And that gene, when it's copied in transcription and then translated in the cytosol, will lead to the production of a protein that can inhibit cell division. So the p53 binds to the DNA. It's a transcription factor. It turns on the gene, and then turning on the gene means transcription is going to occur, producing a messenger RNA. Then the messenger RNA can be moved out into the cytosol so that translation by a ribosome occurs. Then that leads to the production of a protein that can inhibit cell division. Okay, now what that can do is that can allow this process can actually put the cell on ice, so to speak, stop the cell from dividing so that the cell can check its DNA and repair its own DNA. Now, if some of these other DNA checkers figure out that the DNA damage is irreparable, then actually the p53 can signal for apoptosis, or apoptosis, which can basically mean cell death. Now, look what can happen though. Also, if the p53 gene codes for a mutated, if there's a mutation in the p53 gene, it can code for a protein, a transcription p53 transcription factor protein that has the wrong shape. And what that means is it can no longer bind to the promoter at the beginning of a gene. That means this protein, this inhibiting protein, won't be produced. And so that then can certainly mean that cell division is not going to be inhibited because these. So here's a look at some of the different cancers that occur within the U.S. And this is looking back at the occurrences of some of these different cancers and some of the risk factors that are involved in some of these cancers as well.